Hi everyone. This article is regarding the severe acute respiratory syndrome coronavirus, or SARS-CoV, in the early 2000s. The SARS-CoV S protein was generated by vaccinating 10-week-old mice with CMVR plasmid DNA expression vectors. Genes were synthesized through a process of reverse translation. Myeloid dendritic cells were purified from healthy human monocytes, and recombinant retroviruses expressing a bioluminescent reporter gene were produced. Cells were infected with the SARS-CoV strain, and sequential images of live cells were recorded every 3 minutes by confocal microscopy. Bioluminescence assay was performed, and mature myeloid dendritic cells were incubated with the SARS-CoV strain in the presence of a control or an S-specific mouse antiserum. Finally, SARS-CoV envelope proteins were analyzed by co-transfection of expression vectors encoding either the spike, membrane, or small envelope glycoprotein.
In terms of what was found, analysis of SARS-CoV envelope proteins by co-transfection of expression vectors showed that only the S protein mediated entry into target cells. The pH dependence of the SARS-CoV S pseudotyped lentiviral vector revealed the viral glycoprotein entry to be pH dependent. Transduction of various human cell types and lung and renal cells of different species showed that a range of cell types and species are susceptible to infection mediated by the SARS-CoV-S glycoprotein. Renal and pulmonary human cells resulted in high titers of virus, and their susceptibility to pseudovirus transduction correlated to their ability to support SARS-CoV replication. Transfer of the virus was inhibited by a specific anti-S mouse antiserum, verifying that cell-mediated transfer of SARS-CoV is mediated by MDC and is dependent on the interaction of the S glycoprotein with MDC. Relating back to what we have covered in class, this article allowed us to revisit and use our knowledge of glycoproteins and the role they play in viral entry and viral replication. We learned about entry into target cells and the mechanisms of signal transduction, and this allowed us to better understand the results of this experiment in broader terms.
Understanding how SARS coronavirus infects cells is a vital first step before the development of vaccines is possible. This paper was published a long time ago, after the first outbreak of SARS coronavirus in 2003, but its importance and relevance to the current outbreak can be seen by the hundreds of other scientific papers that have cited it, many published recently. Identifying the S protein as the only method of entry for the virus has allowed other researchers to focus their efforts in developing antibodies and vaccines for this protein specifically, since the easiest way to kill a virus is to prevent it from entering the host in the first place. Using this data as a launching point, many different antivirals and potential vaccines are being developed to fight this new outbreak of SARS coronavirus 2. The link to this paper can be found in the description below. Stay safe.